This is lecture 2 on antigens and antibodies. A lot of this will be review for you, but just to make sure that we go over it in the context of blood bank. So antigens or immunogens, they're two different words. Immunogen, technically, if you look up the definition, it says it's anything that can trigger an immune response. It's capable of triggering an immune response. Which means that this is something that's being able to be recognized by the immune system. An antigen is only slightly different. This is something that's capable of binding the product of the immune response, most probably the antibody. If you follow those definitions, an immunogen is an antigen, but an antigen is not necessarily an immunogen. And the reason is they might not share the same epitopes. So an antigen might not be able to trigger an immune response, a certain antigen. It'll be different for each person, okay? But an immunogen, by definition, can trigger an immune response, okay? So since they're not necessarily the same epitopes, they just use a different name. The ability of the antigen or immunogen to produce an immune response, or how strongly it can do that, is called its antigenicity. And that can be different for everybody's immune system, my immune system and your immune system. And that's because it has to be recognized by the genetic markers that you're able to create. The MHC has to be able to bind to, or I'm sorry, the receptors on macrophages have to be able to bind to the antigen so that it can be processed. And then inside the cell, the MHC has to be able to bind to the peptides that are produced. Okay? And so these are different for everybody because we'll have different genetic sequences. Okay? Generally speaking, what we're looking for when we're talking about something that's highly immunogenic is something that has a very highly visible or extremely accessible, is probably a better word for it, epitope. Okay, so if you remember, the most immunogenic materials are proteins. And proteins, you remember, they start out as linear amino acids, but then those amino acids fold up. So if we think of this particular line of amino acids, that the portions that can trigger, that can bind to immune factors are these colored parts here, and they're separated from each other. But when the protein folds, you can see that they're brought together. And this becomes the epitope that binds to the immunological cells, causing the response. Okay, so that is different for every strand of amino acids, and also for every receptor that's produced by you. Okay, it can also be different from one illness to another, but that's a little bit different. The most immunogenic materials are usually extremely foreign. So maybe a sequence of amino acids that's created in horses or in a totally different species. That would be extremely foreign, okay? An unusual phospholipid or an unusual polysaccharide, one that you're not used to ever seeing. That would be something extremely foreign, and your immune system would not be used to it. So it would be more immunogenic. The size also helps to determine immunogenicity. By that we mean the larger the molecule, generally speaking, the more immunogenic, and that is because there are actually more epitopes. So this is the epitope that we colored here on this one, but there's nothing saying that there couldn't be an epitope here on the molecule or here. Any of those things is possible. Okay, so for the most part, for our immune system, the most antigenic epitopes are usually composed of hydrophilic amino acids. They would have to be accessible, obviously, if this is covered over with some other part of the protein. If it's misfolded, it wouldn't be antigenic. But if they're accessible, they're usually a hydrophilic group of amino acids, and that has to do with what MHC can bind as it's being produced in the Golgi. They can be either sequential right in a row, or as you see in this case where they're non-sequential. But as long as they fold to be close to each other, then that's how we get the antigens that the immunogens that we have. Okay. You can have immunogens that are proteins. They're usually the best ones. Polysaccharides, they're still pretty good ones. But the ones that are the least immunogenic are things like DNA, and that's because we all have nucleic acids. So yes, there are sequences that can be immunogenic, but for the most part, DNA, if it comes in connected, in an actual chain, it's pretty much broken down pretty quickly. So the nucleic acids themselves, pretty poor immunogen. And lipids, they're not really all that great immunogen either. If we look at the antigens themselves, or whatever is causing the immune response, we'll just stick with that. I'm going to use the words interchangeably. The antigen itself is usually part of some cell or virus, maybe a toxin or a parasite component that happens to be able to trigger an immune response. Just because, let's say, a strep A, or well, we'll go with an allergen, let's say. Just because peanuts cause an allergy or an immune response in one person doesn't mean that they'll cause an immune response in another. The same thing applies to every other immunogen that's out there. I create an immune response against influenza when I'm exposed to it. But that doesn't mean that everybody's going to create an immune response against that same influenza. They might not have them. You have to have the molecular ability to respond to it. So you have to have a built-in receptor that can bind that antigen. Okay. If you don't have it, then you will not produce an immune response. So immunogenicity is not it's not across all persons of the species. But as a general rule, if we go by general rules, again, the best, the most common things that we see are proteins that made up parts of those organisms or molecules that are processed by antigen-presenting cells and presented to the immune system. They're generally speaking pretty large organic molecules, like, for example, some surface markers that might be present on the organism, maybe some polysaccharides, or not usually lipids, but maybe some polysaccharides that are there as well. So these would be membrane bound or surface markers. They frequently make up the immunogenic portions of whether it's bacteria or viruses or anything else. Again, the more foreign they are, obviously viral markers are more foreign than, in our case, other red blood cell markers. So the more foreign they are, the stronger your response. And when we're talking about red blood cells, those surface markers are the markers that the antigens that we're talking about in blood bank, they're all surface markers on the red blood cell, some of them on white blood cells. And they're supposed to be there. They have a job there. But our body doesn't have the same surface marker on our red cells, and so we respond immunogenetically to them. That's a very fancy word. We respond by mounting an immune response. Generally, the immunogenicity is related to molecular weight. For the most part, the high molecular weight ones greater than 10,000 Daltons will cause an immune response. If you drop below 5,000 Daltons, it's unlikely that you're going to get a response. Okay, only the most hypersensitive might get a response. And then the more molecularly complex it is, also better immune response from that. These are things we take into account when we look at those surface markers that are present on the red cell. There are some very small molecular components, which may, less than 5,000 Daltons, which may be able to produce a response, but they cannot produce a response on their own. They generally need to be attached to some other larger compound in order to get that response. And if you look at this cute little chart here, you can see that, now of course it's upside down. I think that this cartoon is upside down, but it still creates the point. Note that this is the lighter side of the arrow, and so this is, they're showing increasing immunogenicity going down. It's very strange. But if you think about it as maybe the size or the weight, I don't know. Amino acids are very small, and haptens are very small. They have very low immunogenicity, okay? And as you move down, you get to larger and larger molecules, steroids, carbohydrates, and proteins. The immunogenicity increases, okay? So yes, I think this cartoon is upside down. The antigens that we're interested in, for the most part, while we do have to worry about some other ones, the ones that we're mostly interested in are those ones on the red cell membrane, okay? For example, the ones that you already know about, ABO and RH, those are actually portions of markers. They're just small portions of markers that are present for other reasons on the red cell membrane, but they are capable of producing an immune response if they're foreign to you. Okay, so when I take somebody else's blood and I inject it into your veins, it's foreign to you. Okay, I'm trying to get around that foreignness and match it up as close as possible. This is like when you have to take care of somebody's goldfish and it dies and you're trying to go to the pet store to get a matching goldfish. This is what we're trying to do. We're trying to find the matching goldfish in our blood bank. Okay, but these antigens are all actually small portions of molecules that already have a job on the red cell membrane. So we're showing you here the different families in this little cartoon over here, the different families. You have A, B, O, and H. They're part of a marker. It's actually a carbohydrate marker that's attached to the red cell membrane. Purpose of it, we haven't yet figured out. We haven't fully, well, we know actually the sequence and everything, but purpose not necessarily. Then we have other families, MNS, Kel, RH, Diego, and Kid, Duffy. You can see RH, Diego, and Kid, they're actually all found on the same transmembrane protein. Okay, Duffy actually, these two proteins are linked together, and Duffy's pretty much found on the same. You can see it's called a multipass protein. They're closely associated to each other, but anyway, that's besides the point. So when we say that they're all found on there, maybe the RH is found only in this particular spot. Literally, it's an epitope, so it's incredibly tiny. Maybe the Kid is found over here in this spot, over here. And so they're all found in different places on the same red cell surface, red cell membrane protein. So they do have, but they happen to have some other job, which is actually primary. Now the ABO, remember that they probably resemble some kind of environmental element, and that's why we have natural antibodies to the opposite of the antigen that's there. Okay, that has not been completely researched and figured out, and that's because you would have to do a lot of research on fetuses, and there's not a lot of people who want to allow you to do that. But you would have to start out, find when the fetus starts to make those antibodies, and what were they reacting to? Likely it's something that the mother was exposed to and it crosses the placenta. Now, autoantigens are another thing that we do have to worry about in the blood bank. Remember that you're not supposed to, your immune system's not supposed to respond to antigens that you already own. However, there are some mistakes that the immune system can make, can generate antibodies against self-antigens in two kinds of situations. Well, one is where it actually is a real mistake you have a B cell that's able to recognize some epitope that's present on one of your personal cells. Another situation is where your B cells are actually responding to a foreign antigen. Maybe it's an antigen on an EBV virus or something like that. But that antigen happens to match another antigen that is found on one of your cells. Okay, so those are two situations in which an autoantibody would be created against an autoantigen. Okay, and those are something that we do have to worry about in the blood bank. And I'm just going to throw this in here quick just for the last moment on this slide. HLA, or human leukocyte antigen, exactly what it sounds like. These are the surface markers on white cells, in particular on, well, they're actually present on many cells, but HLA is actually major histocompatibility complex, which is in charge of presenting antigen in the immune response. The thing is that major histocompatibility complex is antigenic in and of itself. It has to be able to generate an immune response. That makes sense, right? If it's in charge of presenting to create an immune response, then your immune system has to recognize it, right? It has to be able to recognize it as, oh, that's the guy that's going to tell us what to respond to, okay? So that's the purpose of major histocompatibility factor. The problem is we can have matching blood types, but our HLA doesn't match. And HLA is a lot more complex than ABO. So if it doesn't match, if I happen to get one of your white cells or one of the other cells that express HLA, which includes pretty much every nucleated cell in the body, that would be white cells. Platelets also can do it. If I get one of yours, I might actually produce an immune response against your HLA. And then it can get really complicated. Okay, HLA is actually a lot more complicated than the RBC membranes. But just for at this moment, remember that in the blood bank, the ones that we're interested in, blood group antigens, the red cell antigens, autoantigens, HLA. And just a minor reminder on major histocompatibility complex, it is responsible for presenting antigen. There's two different types. There's class 1 and there's class 2. So this is in here so that you know where it's going to be found. So what are the cells that we're worried about? Class 1, found on all nucleated cells. Remember, red cells don't have a nucleus. No nucleus in a red cell. So it's not on red cells, but it is on every other nucleated cell. Then class 2, that's found on your, oops, sorry, that's a terrible line. That's found on your immune cells. Okay, so you can see DCs are dendritic cells, MQ is another antigen presenting cell, B cells, endothelial cells. So these are your antigen presenting, your immune cells. Okay, and what do they respond to? These are the guys, T cells, CD4 and CD8 positive T cells. CD8 responds to class 1, CD4 responds to class 2. Okay, and then just very generically, we can talk about what kind of proteins, what kind of antigens. Class 1 presents or shows, I should say, intracellular antigens, antigens that come from inside, say, an infected cell. Whereas class 2, that presents antigen that has arisen from external sources. So maybe a bacteria or a virus that was engulfed from outside. Okay, there's a lot of other things here you don't necessarily need to know. You already went through it in immunology. Okay, so this is just a quick one. Know types of cells that they're located on, because you have to know that. Types of cells that respond to it, and then which kind of antigens are present. The HLA itself, the different types, I'm just going to go through this pretty quickly here. You have kind of like the red blood cell types. You have a lot of different HLA genes, a lot more complicated because you can actually carry more than one gene in more than one site. You do carry more than one allele at more than one gene and more than one site. So you have In the class 1 family, the subgroups are HLA A, B, and C. Okay, that's class 1. Class 2, the subgroups are HLA DN, DO, DP, DQ, and DR. Okay, should be easy to remember. Class 1 is 1 letter. Class 2 is 2 letters. There you go. Okay, and then I'm just reiterating here who carries it and what its function is. Okay, remember that you get half of your genes, your alleles, from your mom, half from your dad. But they're pretty much, most of them are controlled by chromosome 6. But because you have all these different genes at these different loci, so you have an HLA A, B, and C. There are different alleles at that loci, and there can be, at some loci there might only be a couple of alleles. In other loci there can be thousands of alleles. So you have a lot more mix here, a lot more variation, and this is where we have the problem with graft rejection, graft versus host disease. We're going to go into a lot more in 2, but this is just a quick reminder how complex HLA is. Now we're going to switch to the antibodies, okay? So the structure of an antibody, okay, to switch to that side of the immune response. Just a reminder that there's just to make sure, just a reminder of the structure here. We have the heavy chains, which are the inside chains here. I'm getting good at writing these lines. Okay, those are the heavy chains. They're identical. You're going to have 2 of them. The light chains are on the outside. Here you go. Okay. Identical. You have 2 of them. Okay. The size is just an interest. You don't really need to know how many, what the size is. But what you can note is that they're approximately, the heavy chain is approximately twice the size of the light chain. Okay. They're held together at a few sites, actually. I'm just going to get rid of these lines. They're held together by disulfide bonds here, here, and then in the middle at the hinge region. Okay, these disulfide bonds are very convenient, and that's because they're bendy. Okay, so they allow the molecule to bend at this, what is called the hinge region. Okay, there's the 2 major parts of any produced antibody include the area here in the circle, in the circles. They're identical to each other, but what they are the antigen binding sites. So this is the site where your antigen, the foreign thing, is going to bind. The other site that's important here is the carboxy terminus. Okay, this is the end of the heavy chain that is recognizable by the immune system. If you have an antigen bound here, then this antibody is saying, hey, this doesn't go here. In order for the immune system to recognize more antigens, we only have to create variety here at the tip of the antibody. The rest of it has to be able to be recognized by the immune system so that you can do something about it. Okay, and so the C3 portion of the antibody is the portion that is recognizable. It's the constant portion. Okay. In addition, there's these structures here. These structures allow you to bind complement, and that becomes important. We'll talk about that in a little bit. Okay. The overall structure of the different classes of antibody. We have 5 different classes of antibody. IgG, IgA, IgM, IgD, and IgE. They're named for Greek letters, gamma, alpha, mu, delta, and epsilon. Okay? Not surprising. Know your Greek letters. Each of those do contain subgroups. We'll get into those in a little bit. Well, not all of them. Some of them contain subgroups. Okay? But what we want you to know is who's a monomer, who's a monomer, and who's a pentamer or a dimer. The IgD, wait, where's my line? IgD, and IgE are, all three of those are monomers. IgA can exist as either a monomer, and it's usually a monomer in the blood, or as a dimer, which is its secreted form. IgM is the largest and is a pentamer, 5 units, 5 monomers bound together. The binding of these monomers together is done in the IgA and in the IgG with J chains. J chains hold each of the units together. It's getting sloppy here. Okay, so that's what you need to note out of this, is the overall structure. There's some other things here as well. The percentage. The largest percentage of any of these in the serum is IgG. 80% of the immunoglobulin in your serum is IgG. The least amount is actually IgE, and that's because most of that is bound to a cell at the time that it's, like, right away after it's produced. And then IgD also very small. Okay, the other two here, this 13% is a little flexible, let's say. Okay, because IgA is generally usually found mostly secreted in the mucous membranes. So you might find this to be much lower in some patients, with the IgM being higher. These two can be interchangeable. Also, I want you to note which one can cross the placenta. There's only one, and that's IgG. Okay. Which ones can fix complement? The two of them can, IgG and IgM. IgA, D, and E do not fix complement. Okay. And so we'll talk about this more, but also biological function. You already know this. Long-term immunity is the purpose of IgG. Secretory antibody for protection, neutralization at the mucous membranes, that's IgA. Initial immune response, that's IgM. IgD is actually a receptor on a B cell. It rarely is involved with directly with the immune response, and IgE is involved with allergic responses and parasites. The heavy chains. Believe it or not, we have different types of heavy and light chains. We have two different types, again, the Greek letters, kappa and lambda for the light chains. And then the heavy chains you already know the names of, G, A, M, D, and E. Okay, you already know them. But what that means is that every time you create an immunoglobulin, you could have the IgG, which means that the heavy chain is a G. So in this case here, the heavy chain is a G, a gamma, and the light chain is a kappa. In this other species, you have a G with a lambda subchain. Okay, these two won't function the same way. They're not this. That's terrible. They're not the same. Okay, so just be aware that you're actually doubling your potential by doubling the types of light chains. The relative amount of immunoglobulin in compared to the rest of the proteins in your body, it can be shown here. This is an electrophoresis, a protein electrophoresis, and it shows the different subunits of proteins present in a normal human serum. The largest subunit is albumin. I don't know if you've seen any of this in chemistry yet, but the largest subunit is albumin. And then we have further subunits, alpha 1, alpha 2, beta, and gamma. Alpha 1 includes things like alpha 1 antitrypsin, TBG, and transcortin. They're normal blood proteins. Alpha 2 includes things like haptoglobin and ceruloplasmin. It also includes the alpha 2 macroglobulin. The beta includes transferrin and beta lipoproteins, and it's the gamma portion here, so named. The gamma portion is where all the immunoglobulins migrate. They go all the way to the end. They're actually pretty small. Okay, and so they bunch up. So this gamma portion is where all G, M, all of those immunoglobulins are contained. Okay, so that's look at the relative amounts. Compared to albumin, significantly less. But if you run a line across straight, which I will not be able to do, it's really not, it's pretty close in the amount with things like transferrin and haptoglobin and alpha-1 chymotrypsin and things like that. The variable and constant regions. So here we have the antigen binding site here and here. And you can see that it consists of 1/4 of the heavy chain, but 1/2 of the light chain, okay? Make that note. So this is the part that undergoes somatic hypermutation, that is produced during somatic hypermutation during the immune response, this variable part, okay? This is the part that's going to attach to whatever's foreign, and it's very specific, okay? The remainder of the molecule has to remain constant. Somebody has to know what's going on here, okay? That's the host binding region. So this is the region, the constant region that your immune cells can bind to, okay? And that's where you have complement activation, which is generally down here on the heavy chain, and then the tissue binding or immune triggering region, which again is your C' region, okay? The structure and function, there is a little bit, this cartoon is just very useful to demonstrate how the hinge region works. Very flexible. Now, there are different types of hinge regions, but very flexible. You can see the different possibilities just based on the structure that you get. You can get a wave, you can get a rotate, you can get a bend, and you can get a wag. So what's the purpose of all this movement? Being able to bind an antigen is one thing, but remember, you need lattice. So in order to get lattice, you need to have all this movement around the structure. The nomenclature, the FAB or antigen binding is what we generally refer to that as, is the top portion. You can see it here. And then the FC, which you might think stands for the constant, the fragment. The F stands for fragment. The C actually stands for crystallizable, and that's because immunologists playing with this stuff were able to cut apart the the antibodies, and they found that this region was able to be crystallized, and later found it was also constant, which was convenient, but not really the point of the C. Okay. Other than that, knowing what the FAB region is and the FC region is, you should be able to say that the FAB region will bind to the foreign material, whereas the FC region will be able to bind to, for example, a neutrophil, a B cell, things like that, okay? So the different subgroups. IgG is the monomer, and again, it makes up most of the immunoglobulin in your serum, but it does contain 4 different subclasses, 1 through 4. Simple enough, okay? They differ for the most part. They do differ in their capabilities, but they also differ significantly in the hinge region. Look at the hinge regions in each of these, and you'll note a big difference, okay? They are all different. They have a different number of bonds in there and stuff. So that's important, and it'll come when we talk a little bit later about the function, the different functions of each of these. IgM, there's only one kind of IgM, thank goodness, but it does have 5 basic subunits. It's the different monomers of an antibody bound together by J chain, okay? Now, because it's so large, it's really not able to diffuse well into the tissues. So if we look in the tissues, in the interstitium between the cells, the lowest amount, we actually 5. Now, that's not the same as the serum, okay? IgM we would find in very, very low quantities in the tissues, just because it really does not penetrate the tissues very well. So it's primarily found in the serum, okay? But because it does have a J chain, it actually can be secreted. The J chain is something that actually is a signal that you can secrete this bad boy here. So because it does have that, you might find some IgM and the mucous membranes, at the mucous membranes, okay? It does have, pardon me, very high avidity, which means avidity is the ability to bind to and hold on to an antigen. It's got a whole bunch of binding sites, so that makes sense, okay? So it's very, it has a very strong hold on its antigen. And also, because it's got 5 monomers, it's got 5 times the amount of complement activation possibility, okay? So you've got a complement activator on each of these 5 molecules, okay? The J chain itself, what is it? It's just a cysteine-rich polypeptide, but that's a sign that it can be secreted. IgA. IgA consists of either 2 kappa or 2 lambda light chains plus 2 heavy chains, okay? It does exist in a monomer and when you look in the serum, what you're testing for is a monomer, okay? However, in the mucous membranes, what we find is the dimer, okay? And that's because that J chain, which contains the secretory piece, that's the piece that signals that it should be secreted, okay? Most other mammals don't have a lot of the monomer. It's secreted almost directly, but they do have the dimer of IgA. And there are 2 subclasses of IgA. We have IgA1 and IgA2. Pardon me. Technically, either one of them, IgA1 or IgA2, can exist in the dimeric form, and so it would be secretory IgA1 or secretory IgA2. So it would have a little S in front of it if it was secretory, okay? Where would you find that? You find it in any mucous membrane. So your saliva, lining your esophagus, your gastrointestinal tract, your eyes, your tears. It also is in colostrum, and it is in the secretions of the genitourinary tract, okay? It's in the blood, it's mostly monomer, okay? There is a chain on the IgA, and you can see it here in brown. You see this chain here, right here. It covers the constant portion of the molecule. Let me get rid of this. That actually helps to protect the secretory IgA from being cleaved by proteolytic enzymes that might be present, because you produce those as well. So if you do produce those, we want to protect the IgA, and so we do that by with that secretory as part of that chain there. In general, IgA is a very poor activator of complement, and it's actually not a very good opsonizer, which means can it trigger on its own a strong immune response? Not really. The strongest immune response can be triggered by IgM. So if IgM binds something, it'll call in the dogs. IgA by itself is not usually incredibly strong, which should make sense. We don't really want to respond to every single thing that comes into our mouth or our gut or our respiratory tract that's bound to IgA. So this will generally trigger a fairly weak response, and we find a lot of IgA against our normal flora, so that we get pretty weak responses there. IgD, again, 2 kappa or lambda light chains along with 2 delta heavy chains. And the function of B cell, this is really just B cell activation and maturation. It's expressed when the B cell exits the bone marrow to populate the peripheral lymphoid tissues. When it reaches a mature state, it'll be co-expressed with IgM and IgD. They'll both be co-expressed. But then once the IgD is, once the B cell is triggered, the IgG is no longer expressed. So it's there basically for in what's considered an immature B cell. IgE is found only in mammals. Its main function, again, 2 kappa or lambda light chains along with 2 epsilon heavy chains. IgE, its main function is in immunity against parasites, worms in particular, and then it has, unfortunately, have this unfortunate side effect that it can be part of allergic responses. But an allergic response is a mistake. Its proper function is to deal with parasites. Parasites like schistosoma or trichinella or fasciola. These are all parasites that you evolved with, and you'd evolved a mechanism to deal with them. Okay. It may have something to do with parasite like malaria, although that has not been demonstrated yet. Unfortunately, because of the way it works, it also does play a role in hypersensitivity, but remember, hypersensitivity is a mistake. Okay, patients who have allergic disease or an allergic reaction, pardon me, you're going to see them producing increased IgE. Okay, and of course, IgE is pretty potent in the immune response that it produces, and so that's why if it's mistakenly triggered, you can see things like anaphylaxis occur, which is an unfortunate mistake. This is just a comparison structurally of the molecules, and you can see here the 5 different subclasses and just the notes that I want you to have on each of them. You have the fact that IgG is the only one that's transferred across the placenta. IgM is the first subspecies to appear after a challenge. IgA is found in the mucous membrane or secretion. IgD, this should really say that it's there during B cell development. And IgE is found on basophils and mast cells. It binds directly to them immediately. So it doesn't function the same way as the rest of them. IgE, it really doesn't circulate. That's not its job. It instead of circulating, once it's made, it immediately binds to basophils and mast cells. So this is kind of like if you think of the antibodies as a defensive response. IgE is a defensive response, but it's bringing the ammunition with it. Okay, it's bringing extra ammunition with it. And if you think about it, when you're trying to respond against a worm, you're not going to be able to engulf a worm. Okay, you can't engulf and process and then present worm antigens. They're too big. So instead, IgE directs basophils and mast cells to the parasite and then just dumps the contents of the cell on the parasite, thus killing it. The function of antibodies is the major one is opsonization, which is the triggering of an immune response. The antibody is supposed to bind antigen in some way. And when it does that, it can do several things. It can neutralize that antigen. So then the antigen can no longer bind. If that antigen, if that portion of the molecule includes, for example, the receptor ligand structures for attachment, then that would neutralize the antigen. That's not always the case, but in some cases it is. The antibody could also cause opsonization, obviously, which is triggering an immune response. It can activate complement, which would then cause lysis of a cell wall, and it can also trigger B cells. To trigger that immunity, that memory, that immune memory. But those are the main functions that antibodies can do. In the blood bank, what we're interested in are the antibodies that are directed against donor cells, okay? And then any donor antibodies that are directed against patient cells. And what we're trying to do is we're trying to avoid these two. We don't want to have patient antibodies directed against any antigens on our cells, and we don't want to have any antibodies in the donor that are going to be directed against the patient cells. Okay, that's our job, is to help avoid that. Okay, and we'll learn how we're going to do that. this is just another cartoon, interesting cartoon to help you note the parts of the structure and the specific biological functions. So you can see antigen binding site here, complement binding site here, macrophage binding site here. This is the stem region. And these are the regions. So on the left side of the cartoon gives you their function, on the right side gives you their regions. Now complement, just a word about complement here. Complement was first discovered by Pfeiffer in 1894. So we've known about this for a long time, okay? But our understanding of it has increased over the century, more than. Both antibody and complement will function as an opsonin. So the antibody can bind to the antigen, to the foreign object. Complement can bind to the foreign object. Your immune system will recognize either one of those, bound complement or the antibody. When it recognizes those things, it'll trigger an immune response, okay? And that'll usually start with phagocytizing the antigen. That's usually the first step. In the case of complement, it could mean engaging the rest of the complement cascade and causing lysis of a cell. In the blood bank, our problem is you have if this is the red cell here, right? I don't know why they made it blue, but that's the red cell, and it has a grossly enlarged antigen on its surface in the form of this yellow thing. Your antibodies recognize it. Fabulous. My antibodies recognize it and bind to it. Okay, now we didn't really want that to happen, right? Here's your donor red blood cell coming in. Your antibodies recognize it. When they recognize it, they will bind complement. That's unfortunate because then this orange stuff is complement. Once you start binding complement, all of the other players join the party, and you end up creating what's called a membrane attack complex, or a MAC, which actually just inserts itself into the wall of this cell here, and they're showing it sort of in 3D here, which causes a change in osmotic balance, and the cell bursts. Okay, binding of antibody can trigger complement, or complement can bind directly to the antigen, and they can be, and it can lead to cell lysis. These things can occur on red cells, white cells, platelets, gram-negative bacilli, any of those, okay? Now we would want it to happen to gram-negative bacilli, not so much to the other three, okay? And the key thing is, once it is triggered, once complement is bound, it's just gonna go the rest of the way by itself. That's what it does, okay? It's a self-perpetuating cycle. Here's the complement cascade. This is blood bank, but you still do need to know your chemistry, okay? The complement cascade it's a whole system of these small proteins that are found in the blood. Some of them are found free in the blood. Some of them are found inside cells and are only released when the cell is activated or infected. They're generally synthesized by the liver, but they circulate in inactive forms or precursors called proproteins. When they are stimulated there are several triggers that can stimulate them. But once they're stimulated, they're going to be triggered to cleave into the proteins. The little proteins are going to be able to cleave, and the two parts that emerge from that include another protease or another complement function, and also a cytokine. So when you have, for example, here you can see C3 here, here we go, is cleaved into C3b and C3a. C3b binds to the surface of the cell. C3a is not non-functional. It actually acts as a cytokine, and it calls in the immune system. And this continues on down the line. So you have C3 cleaving, binding to the red cell. That's going to cause the binding of BB to the C3b, which then, if you have C3 convertase, will convert here. You can see you get C through 4. You get a whole bunch of the C3 with the BB binds to another C3, another 3B, and you get additional binding. The actual cascade I'm not going to worry about in here, but you do have to know that it is a self-perpetuating cycle, and what it leads to, you go C3, pardon me, to C4, to C5, 6, 7, and 8. And once you get this guy in here, C5 bound, then you end up with, and it can be bound to either the C4 or the C3. You see it's bound here as well. Once you get C5 bound, 6, 7, and 8 are going to join the process. That actually is the beginning of your membrane attack complex. Once 6, 7, and 8 are bound, C9 is going to start to bind over and over again to itself. And when it does that, it creates a pore. This is actually a pore in the membrane through that little tunnel there. Okay? And the membrane, and you're going to have the membrane is going to fall under the weight of that osmotic pressure. But what the key thing I want you to realize about these little bits, the C3A, the other bits they didn't show for some reason, but all of them, C4A, all of them have amplification in mind. They're going to call in the immune system, which is going to generate increased complement in the area, increased antibody in the area, increased immune cells in the area. So antibody production over time changes both in subspecies and in the amount, of course. Okay, so if we start if we happen to sensitize our patients, let's say we give them a blood that at some antigen doesn't match our patient. So that would be day 0, right? That's when we give them the blood. Literally in just a few days, you're going to start seeing IgM being produced. You might not be able to measure it yet, but IgM will be the first subspecies that will be produced, and that's shown here by the dotted line. IgG won't come until later, and this chart is very generous at showing it only a week later. It actually can take even longer than a week. But the IgG, if you look, you see that the maximum IgM is compared to IgG on the first exposure. There's a significant difference, okay? Now you see the dip that goes at week 3. You see week 2 for the IgM, there's a decrease. Week 3, you can see there's a decrease in the IgG, and that's because over time, and again, this is just a generalization. In reality, the times are different. But as your exposure to the antibody, the antigen is over, your immune system will stop making so much of that particular antibody. But if you have a second exposure, which they're proposing occurred here, you actually see that IgG takes off right away, okay? And you see a real boost in the response to IgG the second time you're exposed or challenged with an antigen. Yes, you will see an increase in IgM the second time as well, but it's not nearly as significant as the second response to it with IgG. Okay, and that's what we call memory. That's our immunological memory. Now, this is the chart that I wanted you to see that compares the function. Okay, you have IgG subtypes 1 through 4, IgM, A, E, and D. The function of D is, don't worry too much about that. The key things I want you to note in this chart are complement activation. Okay, you can see that IgG3 is the best, of that subgroup, is the best complement activator. IgM, also a good complement activator. The other ones, they're not all that great at activating complement, okay? Neutralization of antigen, opsonization. You can see opsonization, the best ones for that is IgG subclass 1. So opsonization, is it going to be phagocytized? You need IgG class 1 in order to do that, okay? Those are the key things that I wanted you to see on that, but definitely take note of everything on here. Also note, it does have IgA as activating complement. It's actually a very poor activator of complement, and that's the monomer mostly can activate complement. The dimer won't, okay? It's not near it. IgE has nothing there, and that's because it's immediately attached to mast cells and eosinophils and stuff, so it doesn't come into play. The antibody-antigen reactions in vivo, what happens on the patient's body, we try and replicate that in the lab, but we have to understand exactly what's happening in order to know where the differences are. So if you give your patient red blood cells that has those nice surface markers, those antigens that are different from the patient's experience, those markers, ABO, H, RH, all of those can act as antigens in a transfusion situation, okay? So since we are adding, by definition, something foreign, we could potentially cause a transfusion reaction. And what do we see in a transfusion reaction? We see fever, chills, chest pain, back pain, which is where the kidneys are screaming, yelling, or flank pain. Hemorrhage can occur, especially if it's a surgical situation. Increased heart rate because their blood pressure is dropping, shortness of breath, and then the drop in blood pressure. Obviously, we don't want to do this for our patients. Okay, we want to avoid an antibody response to any donor red cell in our patient, which means avoid giving products that have antigens, against which the patient has antibodies. Okay, if you look here in this little cartoon here, this is what you've seen already in the laboratory. You've seen situations where there's no clumping, right? Negative antigen response, positive antigen response. This response, this clumping, this occurs in the vasculature as well. Same thing occurs. So you get clots. The clots cause more vasoconstriction and decreased blood pressure. It really causes a mess, okay? So the agglutination can cause platelet activation. Platelet activation can cause more coagulation. It really can be a mess, okay? And while in the acute one is the one that we fear the most because it's usually much more immunogenic, don't forget that there is a delayed response, a delayed hemolytic reaction that can also occur. In general, if it's delayed, the antigen, the offending antigen, is probably a little less immunogenic, but it's still, at the very least, will shorten the lifespan of those donor cells, which of course is not what we wanted to do. And just so you know how severe the acute reaction can be, they can occur with just a small amount of red cells if it's immunogenic enough. But they don't occur all that often, thank goodness. An acute reaction occurs with the transfusion of red blood cells in about 0.016% of all transfusions. And about 0.003 of those are fatal. So you can see it's a very, very small amount. But we're still afraid that one of our patients is going to be one of those people. It doesn't really help our patient if they're one of them, okay? And the reason is because when a red cell lyses in vivo, all that stuff, all those poisons, all those things that are inside the red cell enter the bloodstream free, and they cause just massive mess, really, okay? So in those cases, that's our worst case scenario. The delayed hemolytic reaction, though, however, that does occur more frequently, significantly more frequently at about 0.025% of transfusions. Okay. Some of them are they're due to the same mechanism. An antibody is bound to an antigen, but usually the consequences are mild. You probably don't have hemolysis. You can have delayed hemolysis, but you probably don't. And if you do, it's probably extracellular. So the symptoms will be a lot less significant. In some cases, patients don't even report a change in their symptoms. In vitro, what we're looking at, again, you've already seen this in a tube. You have a red cell with the little antigens. You want the antibodies to bind to the antigens. You get agglutination if lattice formation occurs, up to and including hemolysis. Okay, both of those are considered positive. And you've already seen those. Those are the things that we look for, that we will be able to predict that this same reaction can happen in our patient. In our tubes, we much more frequently get agglutination, less frequently get hemolysis. The reagents. We just have to talk about the reagents, because the way we see the things that we see are all dependent upon the reagents that we use. Okay, the reagents that we use today are for the most part created using monoclonal antibodies. That's how they create those sera that you know, the anti-sera that you use. So you fuse a mouse myeloma cell with a lymphocyte that produces the antibody of choice that you have, right? And that'll produce a clone, and that clone will continually secrete that specific antibody, making a little biological factory for you. Okay, and that's what we use for reagents. So you stimulate the B cell against the antigen that you're interested in, A, B, D, whatever it is. Okay, then you fuse that cell with a myeloma cell, and you end up with this little biological factory. Okay, the red cell's a little bit different. The red cells, we can't produce those in a mouse. These are actually donors that the manufacturers recruit. They phenotype the donor so they know exactly what antigens are present on their cells. And they have the donors come in on a regular basis to collect their cells for to be cleaned, washed, and then stabilized. We create a cell suspension just like you've used it in the lab. So those come from live donors, but they've been phenotyped. So the real investment that the manufacturer puts in is in the phenotype. After that, it's just phlebotomy. Now, some other little points that as to what's going on in your tubes in vitro is something called zeta potential. Zeta potential is it's a phenomenon that occurs it actually occurs around the red cell that's protective in vivo. It keeps our red cells from sticking together in our vessels unless they're clumped for some reason. But normal flowing red cells should not clump together, and that's because of the electrical charge that's on the surface. They're just ionic charges. But you can see that the negative charges which are closer to the membrane and the positive charges on the outside. So the two positive charges will repel each other. That's good, okay, in vivo. Unfortunately, that can be a problem in vitro because those charges are still there even when we remove the molecules, the red cells from your body. So we don't really want them to repel each other. If we're only talking about an IgM molecule, IgM would be able to cross the difference of the zeta potential pretty easily because it's such a large molecule. But you can see IgG is engulfed by this potential that zeta potential. And so it really on its own wouldn't be able to form a lattice between these two cells unless we can overcome that zeta potential. And we do that mechanically by centrifuging, forcing the cells close together. And we do that chemically by using low ionic strength saline. These are the things. Oh, you can use other things too. What was it? Oh, PEG. You can use polyethylene glycol is another thing that you can use. That will reduce the zeta potential, the charge, around the red cell, so that all it does is bring the cells closer together. If there is IgG, that'll form the lattice. Okay. Another feature that we have to be aware of is something called prozone or postzone. Okay, in order to form cross linkages or that lattice between antibody and antigen, you're actually forming it across two different antibodies or antigens. Okay, so you can see here this antibody is bound to an antigen, but because there's no lattice formed in vitro, we'll never be able to identify this. In vivo, your cells would recognize this part, okay? But in vitro, we would never be able to see this. The same thing on the other side, zone of excess, where you've got too much antigen for the antibodies present. You have lots of antibody bound to, or I'm sorry, antigen bound to these antigens, but very little lattice. There's not enough lattice because all of the antigen binding sites are taken up. There's so much antigen present. We call this the prozone effect. You can call it the prozone effect or the postzone effect. In general, since in vitro we can't tell the difference between these two because we can't see them, we'll call it, I'm just going to refer to it as the prozone. The area where you get a reaction is in the zone of equivalence here, where you have just the right amount of antigen to bind the antigen binding sites, but not too much so that another antibody can bind and you get lattice formation, okay? It's the lattice that you can see. You can't see this over here. Can't see that. Can't see this over here. Can't see that. Okay? How is this overcome? Well, we can overcome this by a couple of ways. The biggest thing being dilution. If you have an antigen excess, you just dilute the specimen and test it again, and you should be able to see lattice formation. An antigen or an antibody excess, that's a problem. You'll never be able to see this one in this particular case. If we don't have enough antigen in the system, you're never going to be able to see it. There's not much that we can do about that. We would miss that if we were testing for it. And then a reminder of the reactions, the phases. Remember what we're talking about here is actually hemagglutination. Recognize the word, okay? Antigen antibody when the antigen is bound to a red cell, okay? This requires a pH of between 6.5 and 7.5. What that means is that you've got to make sure that you've controlled for the pH of all your reagents. The pH of your saline, the pH of all your reagents has to be between 6.5 and 7.5. If it goes outside of that, then your reactions are suspect. We perform these at several different temperatures between 4 degrees Celsius and 37. 4 degrees is not common, but we have that option available. We can do this on a slide or on a tube. Most blood banks do it on a tube, although we do have slide versions of like the ABO. You could do that as well. And we use reagents like LISS or PEG or something in its place in order to deal with the zeta potential. You must be able to grade reactions. You'll learn more specifically in lab how to grade those reactions, but make sure that you're doing it right. You will be tested on it at the end. And you must document the reactions. An inspector can come in at any time they want to see that you documented the reactions. So we have examples here. We have the tube example here. In this case, this is called the solid phase, where what you're looking for is, so the solid phase and the gel are both automated. So you're basically using a microtiter plate here, and in this case, what you have is the antigens on the bottom surface of the well, and you're looking for the antibodies that are present in the patient's serum. Okay, they'll bind to the bottom of the well, or they won't, one or the other. You add the reagents and you get a clump. Here you can see this clump that forms in the end, and that'll be like these little pins on the bottom. You can see that this one is still diffuse, so it's not going this one here is still diffuse. So it's not, you're not going to, the machine is not going to read that as positive. Sloan has the gel technology. We have that too. Gel technology, I think, is a little bit more popular. It's very stable. So as far as documentation, if you were to run a gel on a patient, let's say you were running a gel on a patient and you said, you know what, I'm not really sure how to interpret this. I don't know if I agree with the instrument. Let me hold on to this. And I'll ask my supervisor when they come back from lunch. Perfectly stable. As you know, you would have to respin and reset your tubes, but not the gel. The gel remains stable. But reading it's a little bit different. Basically, at the top of the tubes in the gel, which I think you can see up here. So in this portion of the tubes is where you mix the reagents and the cells. And you allow, the instrument does all the incubation, but you allow the reaction to take place. And if you assume that there is a no agglutination between them, when you spin these guys, when you allow them to settle, the red cells, unagglutinated, will fall to the bottom, okay? And they'll appear on the bottom here of your well. If there is agglutination, if there is lattice formation, they'll get stuck. Maybe right at the top, that would be 4+, maybe a little bit down, 3+, and so on down the line. And you can actually grade that reaction, okay? So that's why gel, people really like it because it allows for the same or similar kind of grading that's possible, which the solid phase is not really as helpful that way. The reagents and the procedures, or what do we use for the what are the parts of the different procedures? In a forward type, you have your patient red blood cell against known antisera. In the reverse type, you have patient serum against known cells. So what are you looking for in those? Well, in the forward type, we're looking for the actual if you're using the patient red cell against known antisera, what you're looking for, what antigens are present on that patient cell. In the reverse, what you're looking for is what antibodies are present in that patient's serum, okay? And they should match, okay? Otherwise, you have a discrepancy. We use AHG in order to detect very weak reactions where basically you just have antibody-coated cells and no lattice formation. When we do that, what we add is AHG, which is basically an anti-IgG that binds to the two antigens, the constant regions of the antigens that are bound, and it causes the lattice formation. So in this case, when we use HGE, we're forcing lattice formation for sensitized cells. Nothing's going to happen if nothing's on that cell, but if there is something on that cell, then AHG will function to form that lattice, okay? And then we use two controls in the blood bank. We use check cells to make sure that our reagents are working. You add check cells at the end in any negative reacting wells to ensure these check cells will definitely interact with the reagents that you have. And so you're just making sure that all your reagents are working. The auto control controls for the patient. And what you're ruling out here is you're making sure that there are no autoantibodies getting in your way. The auto control should be negative. The check cells should be positive for agglutination. If you have a positive auto control, then you have a problem. You have an auto antibody, and you need to get that out of the way. Again, the types of antibodies that we are interested in, or the classes of antibodies that we're interested in the blood bank include, we know that they're antibodies against foreign antigens, in particular blood cell antigens, but there are also different types of responses. You have natural antibodies. Those are the antibodies that you're born with. You start producing antibodies against ABO when you're born. Before you're born with them. The reason for that is not you've not been challenged with anything, but you're still producing the antibodies. So while the understanding is not completely clear, we think that the antigens match something in the environment that triggers that immune response, okay? But it is not the same kind of immune response. Natural antibodies to the ABH blood groups, those are natural antibodies, they're IgM. You don't produce IgG naturally. You'll only produce IgG if you're challenged with a foreign blood group, okay? So those would be called allergenic antibodies. They result only after challenge or sensitization, okay? So whenever I use the word sensitization, it means you've been challenged with something foreign. And these will be both IgG and IgM. Start out with IgM, and then it'll move to IgG. Now, some antigens favor the IgM, some antigens favor the IgG. So we'll go through each one, but it can be either one. Autoantibodies are still a mistake. Your immune system is responding to self-antigens by mistake. They also can be IgG or IgM. And this is just to bring it all kind of together, the total immune response. What happens to our patient? So here's our patient's vessels here. Here's our blood vessel. And we're giving these foreign antigen, red cells, okay? We have a few different ones. What happens? Well, if they're foreign, our antibodies are going to bind to them, and they're going to burst. And you see here, you have hemolysis. Okay, oh, I'm wrong. You have hemolysis. At hemolysis, when all of that hemoglobin is released into the vessel, your body does not like hemoglobin. So you have this molecule here called haptoglobin, which is free in the serum, and it'll bind that hemoglobin and take it for reuse, actually, to be reused in the liver. So that's a good thing. We actually reuse it, but we got to get it out of the vessels first, okay? What happens over time? We got to get rid of that hemoglobin. We got to get rid of the rest of the things that we don't like in there. We're going to clear, I'm sorry. This is just showing, I don't need you to know all this. I'm not going to go through this. But the hemoglobin will be picked up by the haptoglobin and it'll be moved to the liver where it actually can be reused. The red cells, if they're bound with antibody, they'll be removed in the spleen and their life shortened, okay? The key thing to remember here is that we have an awful lot riding on the work that we're doing. We want to make sure that we get it right, okay? Because we don't want to cause this reaction here. We also don't want to cause a senescent red cell so this is a red cell that's coated with antibody. It becomes useless to our patient. It's going to be removed in the spleen, in the liver too, mostly in the spleen. And so you're not going to benefit from it, which means you're going to increase the oxygen stress, the problem that you have going on in the patient's tissues. So be sure, watch what you're doing, follow the procedures closely in blood bank, everywhere, but in blood bank we're particularly sensitive to it. And then just a reminder, number one cause of mistakes in the blood bank is actually clerical. So I just want to bring you back to this, okay? There are so many steps along the way that have to be, that we have to ensure are correct to ensure that our patient, way down here at the end, gets the right blood, okay? Those include recruiting donors, screening the donors, collecting and preparing the unit, including infectious disease testing. That's before we even know who's going to get this, okay? Then when we have a patient who needs blood, making sure that the correct patient sample is drawn. Wrong blood in tube is number one problem, okay? Making sure that it's medically necessary for the patient to receive this blood, okay? That all of our testing is correct. And then even after it's released from the lab, is it delivered properly? Is the temperature kept right? Is it administered properly? Blood type right? All that fun stuff. There's tons of steps along the way. And we have to make sure that we watch every single one of them, and we're constantly looking for ways to reduce that clerical error to 0.